Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Dr. Rizwan here from the Department of Biochemistry, Independent Medical College, Faisalabad. Today we will discuss our new chapter, that is the cancer metabolism. The objective of this chapter are to describe the important properties of the oncogenes and tumor suppressor genes, and briefly describe the concepts of genomic instability, aneuploidy, and angiogenesis in the tumors. The term cancer, it is derived from a Latin word, that is the cancrum, which means a crab. The disease is so called because the swollen veins around the area it resembles a crab limb. Now we'll start from the biomedical importance of the cancers. The cancer cells. The cancers constitute the second most common cause of death after the cardiovascular disease. Approximately 8 million people around the world died from the cancer each year, and this figure is projected to increase in number. Humans of all the ages develop cancers, and a wide variety of organs are affected. Worldwide, the main types of cancer accounting for mortality are those involving the lung cancer, the cancer of stomach, the cancer of colon, rectum, cancer of liver, and the cancer of breast. The other type of cancers that lead to the death includes the cervical, esophageal, and the prostatic cancers. The skin cancers are very common, but apart from the melanomas, are generally not as aggressive as those mentioned above. The incidence of many cancers increase with the age. Hence, as the people live longer, many more will develop the disease. Here in this slide, we will discuss some general comments on the neoplasms. A neoplasm refers to any abnormal new growth of the tissue. It may be benign or malignant in nature. The term cancer, it is usually associated with the malignant tumors, and the cancer cells, it is characterized by certain properties which are discussed as under. Number 1 is the proliferate rapidly. They display diminished growth control. They display loss of contact inhibition in vitro. They invade the local tissue and they spread or metastasize to the other organs or the other parts of the body. They are self-sufficient in its growth signals and are insensitive to anti-growth signals. It stimulates the local angiogenesis and are often able to evade the apoptosis. These are the key points and the properties of a typical malignant tumor cells. It is metastasis that generally responsible for the death of the patients who develop the cancers. In this slide, we will see on the left side these are the 6 major features of the cancer cells which we have discussed earlier. In the right side of the picture, these are some biochemical and genetic changes that occur in the human cancer cells, and the many changes in addition to those which are indicated are observed in the cancer cells, like the role of mutations in activation oncogenes and inactivation tumor suppressor genes, the abnormalities of the cell cycling and the chromosome and the chromatin structures, including aneuploidy, the alterations of the expression of the specific messenger RNAs and the regulatory nuclear RNAs have been reported, and the relationship of the stem cells to the cancers. The tumor sometimes synthesize certain fetal antigens which may be measurable in the blood, and the changes in the plasma membrane constituents. Many changes in the metabolisms have been observed. For example, the cancer cells often exhibit high rate of aerobic glycolysis, and the other properties are the cell adhesion molecules, and they also affect the extracellular matrix. The fundamental features of carcinogenesis: there are the four classes of the genes. Number one is the proto-oncogenes, tumor suppressor genes, DNA repair genes, and the gene regulating apoptosis. When the mutation occurs to these genes, or a loss of the function or inappropriate regulation, they can result in development of a tumor. The cancer of the colonal origin with a single abnormal cell, often with the multiple genetic alterations, multiplying to become a mass of a cells forming a tumor, which can cause the genetic damage to the tissue, and could be familial and spontaneous mutations which may predispose to formation of a cancer. There are the three classes of carcinogens. These are the radiant energy, chemicals, and certain viruses, exposure to which results in the tumor formation. The radiant energy and the chemicals, they cause mutation in DNA, and the third class generally act by introducing a novel genes into the normal cells. The radiant energy can be carcinogenic. The ultraviolet rays, X-rays, and the gamma rays are mutagenic and carcinogenic. Extensive studies have shown that these agents can damage DNA in number of ways, including the lesions and exposure to the ultraviolet radiation. It is common due to the exposure to the sunlight, which is its main source. The risk of developing the skin cancers due to the ultraviolet radiation increases with the increasing frequency and intensity of exposure, and decreasing the melanin content of the skin. Secondly, the many chemicals, they are carcinogenic. The most of the chemical carcinogens are thought to covalently modify the DNA, thereby forming a wide range of nucleotide adducts. Depending on the extent of the damage to the DNA and its repair by DNA repair system, some chemicals interact directly with the DNA. For example, the methchloroethamine and the beta-propionolactone. The term procarcinogen requires the conversion by the enzyme action to become ultimate carcinogens. The chemical carcinogenesis comprises of the two stages, that is the initiation and promotion. In its first stage, that is the initiation, is a stage where exposure to a chemical causes irreversible DNA damage and is necessary initial event for a cell to become cancerous. The second stage is the promotion. It comprises the stage at which the initiated cell begins to grow and proliferate abnormally. The cumulative effect of these two stages is resulting in the formation of a new plasm. There are approximately 15% of the human cancers. It can be caused by different viruses. The study of the tumor viruses has contributed very significantly to understanding the spread of cancer. In general, the genetic material of the virus is incorporated into the genome of a host cell. And in the case of RNA virus, this would occur after the reverse transcription of the viral RNA to viral DNA. Such integration of the viral DNA, it is called the provirus, with the host DNA results in various events such as deregulation of the cell cycle, inhibition of apoptosis, and abnormalities of the cell signaling pathways. The DNA virus often act by downregulation the expression and the function of tumor suppressor genes and their protein products. On the other hand, the RNA virus often carry oncogenes in their genomes. In this table, we can see the different viruses that cause, which are associated with the human cancers. Like Epstein-Barr virus, it is a DNA virus, and it is responsible for developing the Burkitt lymphomas, nasopharyngeal cancers, and beta cell and B cell lymphoma. The second is the hepatitis B virus. It is again the DNA virus. Hepatitis C, it is RNA virus, which can lead to the hepatocellular carcinoma. The human herpes virus type 1, it is again the DNA virus. It can lead to the formation of Kaposi's sarcoma. Human papilloma virus, it is again the DNA virus, which can lead to the formation of the cancer of the cervix. And human T cell leukemia virus type 1, it is the RNA virus, which can cause the adult T cell leukemia. In addition to all these viruses, in 2020 we have come across a very serious, that is the COVID-19. It is again the RNA virus, which can lead to the formation of this deathly disease, that is the corona. The oncogenes and the tumor suppressor genes, they play a key role in causing the cancers. The two key findings were the discoveries of oncogenes and the tumor suppressor genes. The oncogenes and the loss of activity of tumor suppressor genes, they derive the cell growth, which can lead to development of a cancers. The oncogenes encode various proteins that can derive the growth of a cancer cells. The oncogenes are derived from proto-oncogenes, and the tumor suppressor genes encode the proteins that normally suppress the cell growth, but which are inactivated when altered by mutations. The microRNA molecules are also affected by mutation, and this can affect their normal regulatory functions. In addition, the epigenetic changes affect the genes expression, and hence growth of a cancer cell occurs. The oncogenes, they can be defined as an altered genes whose product act in a dominant manner to accelerate the cell growth or cell division. The oncogenes are generated by activation of normal cellular proto-oncogenes, that is, the genes encoding the growth stimulating proteins. Such activations can affect through any several different mechanisms, and these mechanisms are shown in this table. These mechanisms could be the mutation, it could be the promoter insertion, it could be the enhanced insertion and the chromosomal translocation and the gene amplification. Now I'll discuss the chromosomal translocation. The reciprocal translocation is seen in the Burkitt's lymphoma. The chromosome involved are A8 and 14. A segment of the Q arm breaks off and moves to chromosome 14. A reverse process moves a small segments from Q arm of chromosome 14 towards the chromosome 8. The Mike gene is contained in a small piece of a chromosome 8 and is transferred to chromosome 14. It is thus placed next to a gene transcribing a heavy chain of immunoglobulins molecule, and it becomes activated. Here are some properties of important oncogenes and the tumor suppressor genes like Mike genes, P53, RAS and RB genes. The Mike is a family of regulatory genes. It is an oncogene that encodes DNA binding transcription factors. P53 that can alter the transcription which involved in the cell growth, cell cycle, progression and the DNA replication. The P53 genes, it is a tumor suppressor genes that responds to a various cellular stresses. It includes the cell cycle arrest, apoptosis, DNA repair, and is involved in some aspect of regulation of the cellular metabolism. The RAS, it is a family of oncogenes encoding small GTPase. They were initially identified as being the transforming genes of certain murine sarcoma virus. The RB genes, also known as the retinoblastoma genes, it is a tumor suppressor gene encoding the retinoblastoma proteins. The RB genes regulate the cell cycles by binding the elongation factors E to F, and it represses the transcription of the various genes involved in the S phase of the cycle. We will end up over here, and in the next lecture we will start from the different growth factors and abnormality of the receptors and signaling pathways which play a major role in the development of a cancer. Okay students, Allah Hafiz.